My name is Bin Chen. I'm from the Department of Pediatrics and Human Development, and also the Department of Pharmacology and Toxicology.
Hi, my name is Shreya Paithankar. I work as technical assistant at Dr. Binchin's lab.
So we are using big data and artificial intelligence to discover new therapeutics for COVID-19 through this collaboration with a bunch of scientists and the ICU physicians. In particular, we are interested to identify small molecule, including FDA-approved drug that can target the host cells. To target the host cell, many studies right now are trying to identify small molecule to modulate one single protein. But as we all know, the viral infection involves multiple processes. It would be great if we could identify small molecule to modulate multiple biological processes. So we are using gene expression to represent the multiple biological process and trying to develop a new way to identify small molecule to reverse the global gene expression. So using this approach, we are able to screen all the FDA-approved drugs and look at whether they have a potential to be used to treat the COVID-19. Meanwhile, we can also discover new molecules, small molecule, to reverse the gene expression of the COVID-19 infection and Pfizer to identify new therapeutics. At MSU, we have very limited resource for the COVID-19 patient. However, fortunately, there are lots of data publicly available. Even before the publication, researchers already published their data to the public repository that allow us to use them. And the second challenge is that to validate our hypothesis. Once we identify the drugs, at MSU, again, we also have very limited resource to access to the virus. So at the very beginning, I sent a bunch of the cold email to the scientists across the world to ask whether they would be interested to test our candidates. Fortunately, 2 labs expressed their interest to work with us. So now we are working with them to validate our drug candidates. We fortunately identified about 10 candidates, and we saw their efficacy in vitro model. Unfortunately, they're also toxic. So now we are developing a model to be able to predict their toxicity using existing data. So that's one direction. And also we are trying to work with a bunch of hospitals to validate our hypothesis using electronic medical records. We want to look at whether this drug will be effective in patients in the real world data. And the third, by collaborating with our computer science faculty, Professor Jiayu Zhou, we developed deep learning method to be able to screen a large amount of compounds for the COVID-19. So we have 3 main directions.
Our lab is a big data lab. We always start our research based on the data available in the public repository, and we try to find out maximum and useful data regarding the SARS-CoV-19 virus. So initially we got data which was not for the SARS-CoV-2 but the previous SARS viruses, in which they have actually infected the virus to they are infected the cell lines with this virus, and to see how the cell lines are reacting to the virus infection. So all this data we download from the NCBI SRA to our HPCC account, and then we have inbuilt RNA-seq pipeline to process all the data. So we process the data on HPCC.
At MSU, we have only very limited resource to access to the patient data, but there are lots of data publicly available we can leverage. At MSU, we are very strong engineer and also the competition infrastructures. We can leverage our strengths to help the patient. For the downstream analysis or validation, just don't be shy. Go out if you have a great idea. Because this is a global effort, I think many of the scientists are willing to help.